Hello, everyone. Thank you very much for attending this talk today. I am Anne-Sophie Payas-Jimenez, Head of R&D at Angle Europe Limited. We're based in Guildford, UK. Today, I will present the development of a standardized assay that allows for PD-L1 detection on epithelial and mesenchymal CTCs using the Parsortix system. First, let me introduce a little bit Angle. So Angle is a CTC liquid biopsy company. And our mission is to enable personalized cancer care by providing the complete picture of the patient's cancer from a simple blood test. We've got 3 different locations. So as I said, one in Guildford in UK and 2 other ones, one in Canada in Toronto and another one in US in Pennsylvania. So why is it important and interesting to look at liquid biopsy? Liquid biopsy is a non-invasive technique and allow for repeat samples. From the samples, you can have a large range of information. I gave you here a few examples. So for example, cell-free DNA, circulating tumor cells or other immune cells, but also some extra vesicles, for example, or soluble protein. It has been published that liquid biopsy enabled to reveal undiagnosed tumor heterogeneity. If you look in detail of CTC, so circulating tumor cells, so as I just said, that's allow for non-invasive diagnostic. And it has been published as well that CTC reflect the metastatic tumor. From CTCs, like all other cells, you can have access to protein, nucleic acid, and a lot of different information that give you a huge source of biomarkers. However, CTC are rare. So you can have 1 CTC out of 10 to 8 blood cells. So it is crucial to have standardized CTC enrichment technique. At Angle, we commercialized an instrument which is called the Parsortix technology, which is a platform that allows for harvesting rare cells from a liquid biopsy sample. So here, for example, in this talk, I will focus on circulating tumor cells enriched from a blood sample. So how does it work? So the Parsortix technology is an epitope-independent technology. It works based on physical cell property and cell size. I will explain you a little bit more. So on the left-hand side, here you've got a picture of a Parsortix. So a Parsortix is a bench size instrument. And on the left-hand side here, you've got a schematic representation of a Parsortix cassette. So here on the plan view, it's the cassette. So the blood is processed through this microfluidic instrument, the Parsortix, and come here through the inlet in the bottom, go through the cassettes and get out by the outlet. But how does it actually work? So each single line represented here is actually a series of steps that end by a final gap. So you got here a cross section. So you can see the final gap, the different steps here. So we've got 10 different steps that end by a final gap here. The final gap is 6.5 micron. The blood cells, the blood components, are very often smaller than 6.5 micron, or at least they can squeeze through this final gap because they are highly compressible. However, CTCs are often bigger than 6.5, but also less compressible than blood cells. So they get stuck just before this final gap. How to recover the CTCs? So it's very simple. The instrument inverts the flow and run buffer from the outlet to the inlet. And then you can collect the enriched fraction in buffer. So the Parsortix technology can actually capture a broad range of viable CTCs as well as CTC cluster. At Angle, we developed and we are developing several workflows that give a range of research possibilities. So let me go through this generic slide. So as I said, from a blood sample, so it could be different blood sample. It could be fresh, like blood collected in EDTA, but also preserved or stabilized sample. So here, for example, I mentioned Streck tube, but it can be a lot of different blood tubes. So once the blood is collected in this tube, they go through the microfluidic system at the Parsortix. And as I mentioned earlier, you've got a fraction of CTC clusters, and immune cells. But what to do with those cells? So first, those cells are viable if, obviously, if the blood is collected in EDTA, you can use those cells to culture or to do some xenograph experiments. But you can also look at what the biomarkers and what are on those cells. So for that, you can do some immunocytology assays to look at protein or RNA, but also you can do some molecular analysis and look at the RNA. So today I decided to focus my presentation on the development of an immunofluorescent assay. We look at PD-L1. So PD-L1 allows cancer cells to evade the host immune response when upregulated. It is very important to look at the PD-L1 expression because the measurement of this expression in circulating tumor cell may enable repeat testing to provide up-to-date PD-L1 status, and then give the potential to monitor patients that are on these therapies. So let's evaluate the performance of a research use only assay for the characterization of PD-L1 expression on epithelial and mesenchymal CTC isolated using the Parsortix system. Let's go back to the workflow. I will give you a little bit more detail. So here in this study, we processed Streck blood samples, so blood collected in Streck tube, for up to 120 hours after blood collection. The blood has been processed using the Parsortix instrument, and the CTCs and cell has been enriched. Once enriched, so the cells have been stained using an immunostaining. So here we use different markers. So first, nucleus, obviously, to detect nucleated cells. But then we combine 4 different set of markers. So one set that will target epithelial cells, one that will target mesenchymal cells, obviously your biomarker, so here PD-L1. But then on Angle, we actually develop positive and negative selections within the same assay, which means that you will have in one side a positive selection with epithelial and mesenchymal markers, and another side blood cell markers that which allow you to detect blood cells. Once the cells are stained, we image and analyze them using the BioView. So the BioView system will give you a gallery of images that you can actually review, and also give you the analysis by categorizing the cells regarding their expression, their gene expression. So we developed this assay using cell lines first. I wish I could go in detail to this development, but unfortunately I don't have time, but I will briefly go through this. So as I said, we use a combination of different markers. So here is a 5-color assay. So one color is for nucleated staining, nuclear staining in blue. Then we've got one channel to detect epithelial phenotype, another channel to detect mesenchymal phenotype. So you can check which cells are actually epithelial, which cells are mesenchymal, or which cells are actually undertaking the epithelial mesenchymal transition or mesenchymal epithelial transition. Then you've got one channel which is free for your biomarker, so here PD-L1. And finally, a 5th channel, as I said, to detect blood cells. So to have specific marker for blood cells and give you this positive negative selection that I mentioned earlier. So I give you here a few examples. So you can see on the left-hand side on the top here, an epithelial cell here that express PD-L1. On the left-hand side, but bottom one now, you've got a cell which is an EMT, so or MET, so mesenchymal epithelial phenotype that also express PD-L1. Here, an EMT cell that doesn't express PD-L1, and here a cell that doesn't express doesn't express epithelial or mesenchymal marker, but express PD-L1. This cell is not counted as a CTC in our assay. So in order to evaluate the performance of our assay, we got access to commercialized cancer patient samples. We actually process 8 healthy volunteer, 16 metastatic breast cancer patient samples, and 20 non-small cell lung cancer patient samples. Out of the 16 metastatic breast cancer patient sample, 8, so half of them, show at least one CTC, and the range is going from 2 to 63 CTCs. In non-small cell lung cancer, out of the 20 samples that we processed, 30% show at least one CTC, and here the range is coming from 1 cell up to 80 cells per blood sample. We didn't detect any cell in healthy volunteer. It has been published recently or less recently that clusters are a huge source of information on the metastatic status of the patient. So we looked as well at the cluster, and we show that 90% or a little bit less than 90% of the metastatic breast cancer patient sample show at least one cluster. And the size of those clusters goes from 2 cells to up to 14 cells. And we can detect from 1 cluster per donor up to 11 clusters per donor in this cohort of metastatic breast cancer patient samples. In the non-small cell lung cancer patient samples, 2/3 of those patients that got at least one CTC show at least one cluster. We got from 1 cluster per donor up to 6 in this cohort, and the clusters go from 2 to 74 cells within 1 cluster. So there is a significant difference between the healthy volunteers. As you can see, there is no CTC detected, and the cohort of patient samples here where we detected at least one CTC. But let's look at the PD-L1 expression on those cells. So as I said earlier, 50% of the metastatic breast cancer patient samples that we process got at least one CTC. And out of those 50% samples, 1/3 of them show PD-L1 only cells. So all the CTC detected on those patient samples are PD-L1. And 2/3 of those samples show at least one PD-L1 positive cells, CTC. In the non-small cell lung cancer patient sample cohort, as I said earlier, 30% show at least one CTC. And here, 2/3 of the patients show PD-L1 only CTCs, so which means that all the CTCs in the 2/3 of those patients show a PD-L1 positive status. As a note here, all the PD-L1 positive cells were expressing or epithelial or epithelial and mesenchymal transition phenotype or mesenchymal only marker. I give you here an example of a cluster. So you can see here, you can see here a cluster of cells. It doesn't express the blood cell markers, but are epithelial for most of them. Here you can see one is not, for example, but this one is mesenchymal. All of them in this cluster show a mesenchymal phenotype. And most of the cells in this cluster show PD-L1 expression. One here, for example, doesn't show PD-L1 expression. So you got here an heterogeneous cluster showing PD-L1 expression for some of the cells, and some of the other cells doesn't express PD-L1. Some other examples. So here you got another heterogeneous cluster, but this one is heterogeneous because here in the middle, you can see there is a blood cell. And you can see here a blood cell marker signal in the middle. Here it's empty. All the other cells express epithelial markers, quite weak here. Strong mesenchymal markers, staining, and PD-L1 expression. And here the same with a smaller cluster. So as conclusion, I hope that I convinced you that the Parsortix technology can harvest CTCs and CTC cluster from a blood with a high recovery rate, including epithelial and mesenchymal CTC and CTC clusters. I show you here some example again. So different biomarkers are being used to CTC identification and characterization using immunofluorescence assay. So here I gave you an example of PD-L1, but we can actually develop the same kind of assay with a lot of different biomarkers. There's time for me for the last minute of my talk to introduce Angle Pharma Service Lab. So Angle recently opened 2 service labs that are GCLP compliant, one in US and one in UK, and are backed up by extensive expertise in clinical lab operation and CTC. So those 2 service labs provide global support to pharma studies by we offer enumeration of epithelial and mesenchymal CTCs and CTC clusters. We also offer the assay that I presented today, which is looking at epithelial mesenchymal CTC and CTC clusters, as well as PD-L1. Currently under development, we've got another assay, which is looking at enumeration of epithelial and mesenchymal CTCs and CTC clusters, but also at DNA damage repair markers. And as I briefly mentioned earlier, we can custom your assay. So we've got a service which is dedicated to custom assay development. and that will work with you to add any additional desired marker to our epithelial mesenchymal assay. And in 2022, we will be able to offer a multiplex gene expression analysis. So we've got assays that can cover up to 100 genes for prostate, ovarian, or breast phenotype. And we also got a custom gene panel within this multiplex gene expression assay. So thank you very much for attending this talk again, and I will be more than happy to answer your question in the Q&A live session. Thank you.